Hello students, welcome back to Trendy Achievers channel. Today we are going to discuss a very important concept for second year BPC students in the case of botany, that is glycolysis pathway. Glycolysis means the breaking down of glucose to two pyruvic acid molecules. So here what is meant by respiration? First of all, respiration is the breaking down of sugar to give rise to energy. So here the glycolysis is the partial oxidation of glucose molecule to give rise to 2-pyruvic acid. So these 2-pyruvic acid molecule, these enter into one more cycle, that is TCA, tricarboxylic acid cycle. So here in Greek, glycose means sugar. So that sugar, so the carbohydrate, sugars are nothing but carbohydrates. Here the glucose and fructose are the monomers. They derive from the carbohydrate source, that is sucrose. From sucrose, with the help of the enzyme invertase, glucose and fructose acts as a they enter into the glycolysis pathway. So here the glucose molecule, it is a 6-carbon compound. So this glucose 6-carbon compound molecule, a glucose molecule, by the process of this glycolysis, it is very important for long answer question. So by this pathway, you can easily remember. If you watch my video from starting to ending, I'll be giving you a very easy and important steps which involved in the glycolysis. It requires nearly, it occurring 10 steps. Okay. What are those first? What are the steps? What are the enzymes? What are the products in each step? We are discussing. It is very important for a long answer question and even MSET and NEET point of view also very important. Okay students, let us enter. So first step is the phosphorylation. By the process of phosphorylation, glucose molecule, it converts into glucose 6-phosphate by the process of phosphorylation. Phosphorylation means the addition of phosphate. So here the ATP molecule, one ATP gives rise to ADP. So that phosphate utilized for the conversion of glucose to glucose 6-phosphate with the enzyme hexokinase and Mg+2 ions. So these glucose 6-phosphate converts to fructose 6-phosphate. These two are isomers with a phosphohexose isomerase. So it is also 6-carbon compound. The process is isomerization. So isomerization nothing but the rotation, just changing. So this glucose converted to fructose 6-phosphate. Fructose 6-phosphate converted to fructose 1,6-bisphosphate with the enzyme phosphofructokinase and magnesium ions. And here also phosphate required, so that phosphorylation. Here also ATP converted to ADP. Next, fructose 6-phosphate converted here, it undergo cleavage process. Cleavage means breakdown. 6-carbon compound breakdown to 3-carbon compounds, that means 2. One is dihydroxyacetone phosphate and glycerol head 3-phosphate. So these two are 3-carbon compound. So with the enzyme aldolase. So fructose 1,6-bisphosphate converted to this cleavage, undergo cleavage. DHEAP as well as glycerol head 3-phosphate, they both can rotate. So that process is called as isomerization. With the help of triose phosphate isomerase enzyme, they can convert DHEAP to glycerol head 3-phosphate. So these glycerol head 3-phosphate, here it undergo oxidation and phosphorylation. So here there is oxidation with the help of glycerol head phosphate dehydrogenase. Here the hydrogens are removing and converting to adding hydrogens to NAD to NADH plus H plus. So here oxidation takes place. So these next glycerol head 3-phosphate undergo oxidation and phosphorylation and produces 1,3-bisphosphate glycerate. 1,3-bisphosphoglycerate. So here it is the wherever the NADPH NADH produce, it is the oxidation and phosphorylation undergo. So next 1,3-bisphosphoglycerate undergoes 2 molecules of 3-phosphoglycerate. What producing students? 1,3 from 1,3-bisphosphoglycerate to 2 molecules of 3-phosphoglycerate. So here it requires, so here the ATP producing. So that ATP is producing from the substrate. So that we call this process as substrate level phosphorylation 1 or dephosphorylation. So here ADP converted to ATP1, ATP1 phosphate is adding. So that this process is said to be substrate level phosphorylation. So the enzyme here required is phosphoglycerokinase and magnesium ions. So next, these 2 molecules of 3-phosphoglycerate undergo isomerization into 2 molecules of 2-phosphoglycerate. So just change the structure with the help of phosphoglyceromutase enzyme. Next, these 2 molecules of 2-phosphoglycerate converted to 2-phosphoenol pyruvate by the process of removing water molecule. That process is called as dehydration with the enzyme enolase and magnesium ions. Next, these 2 molecules of 2-phosphoenol pyruvate converted to 2 molecules of pyruvate or pyruvic acid in the presence of enzyme pyruvate kinase, and the process is called dephosphorylation. Here also ATP is producing. So it is also producing from substrate, so that it is called as substrate level phosphorylation too. Okay students, once again I am repeating the steps. First is phosphorylation, second is isomerization, third is phosphorylation, fourth is cleavage, fifth is isomerization, sixth is oxidation and phosphorylation, seventh one is dephosphorylation or substrate level phosphorylation 1. 8th is isomerization. 9th is dehydration. 10th is dephosphorylation or substrate level phosphorylation 2. So these enzymes and this first you should remember steps students. So next, what is converting glucose to? So what the sequential steps were identified or discovered by 3 scientists. They are Emden, Meyerhoff and Paranas. These 3 scientists observed the sequence or reactions that occur in the glycolysis. So that glycolysis pathway is also called as EMP pathway. It is very important for long answer questions. Okay students, I hope it helps you a lot to score out of Maths in final exam. Thank you.